Hello, and thank you for joining this presentation entitled Decoding Human Fetal Liver Hematopoiesis. My name is Graham Milton, and I am a European Market Development Manager for Immunology Research at STEMCELL Technologies. This is a recording of a presentation given by our guest speaker, Dr. Rachel Botting, during a Lunch and Learn event at STEMCELL's Cambridge, UK facilities. Dr. Botting studied biomedical science at the University of Technology of Sydney, including gene therapy targeting liver metabolic diseases at the Children's Medical Research Institute. She undertook her PhD studies at the University of Sydney at the Westmead Institute, studying the sexual transmission of HIV. Dr. Botting is currently a research associate in Professor Hanifa's lab at Newcastle University, which specializes in using single-cell technologies to investigate organs and systems during development, health, and disease. During this presentation, Dr. Botting shares with us her lab's research from a recent publication. Please enjoy the presentation.
Thank you very much. So yes, as was mentioned, I'm going to go through some of the work that we've been doing in the Hanifa lab, specifically looking at one of our more recent publications, which was looking at decoding human fetal liver hematopoiesis. So this was a big collaborative effort. It was mainly led by our lab, but we did a lot with the Sanger Institute, and then we did some comparisons with data sets from overseas, so there's quite a few that were involved in this study. So the reason we wanted to look at this is we wanted to look at the early blood and immune development. So this is really critical for survival and health of the organism. So seeding the immune system very early on, and then obviously how that progresses through into adulthood, the comparisons that we can see there, and all of it interacting together. So basically what you get is, starting on the left, you've got the yolk sac, and that development goes through, and you start picking up the AGM starts to develop some of the hematopoietic development, which goes through to the fetal liver that largely takes over for the rest of gestation. And the bone marrow starts to kick in. It's seeded around 11 post-conception weeks, and then it's not until around 16 weeks or after that the bone marrow really starts to pick up, which is the blue. So, sorry, you've got that's the yolk sac. The liver's in red coming up, and then the bone marrow's a bit later on. So one of the things that we've found when you look through a lot of what is known about early blood and immune development is it's reliant on model systems. So quite a bit is done in mouse, which is really nice in that, and it allows the in vivo and I guess more long-term studies if you're trying to do tracing. But there are quite a few differences between mouse and human. So we know that there are different cell types there, the timing of things are different, so depending on what immune cell type you're looking at, they come up at different stages during development comparatively, or even the organs that are being developed. So what might be happening, for example, in the thymus of a human is not timed the same as what's happening in the thymus of a mouse. And so really, in the broader scheme of things, is how does this translate to, I guess, various diseases that we know have a fetal origin, so things that are congenital and come up very early after birth, so looking at anemias, various different primary immunodeficiencies and infant leukemia, and also what comes through into adult pathology, so especially in cancers, some of them, they kind of revert to more of a developmental state in losing their checkpoints and things like that. So when do things switch over, and how might you be able to switch them on? And then looking at how you might be able to manipulate the stem cells that you find in fetal for various therapeutic options. Really, this was the brainchild of my boss, Ms. Hanifa. And we do all of this under the umbrella of the Human Cell Atlas, and then more within that, the Development Cell Atlas. So it's a huge initiative that's trying to map all of the cells in primarily the healthy human state, but then also going on to look at diseases. And it wouldn't be possible without the HDBR, so the Human Developmental Biology Resource. They kind of cover all of the ethics, they do all of the dissections, everything like that. So we really rely on their expertise to be able to get the tissues to do the work that we're doing. So what we did is we looked at, liver was the primary interest, and then we looked at kidney and skin from the same sample so that we could see how the cells were seeding out into non-lymphoid tissues. And we also got yolk sac to get some of the early. All of our dissociations were done with digestion, so we do collagenase digestion to get the cells out. We do a FACS enrichment, We did that, so we still did it in an unbiased way if we ran both fractions to make sure that we captured everything. But the CD45 positive fraction is a lot smaller in most of the tissues. In skin, it might only be, say, 3%. So you don't get the diversity if you run it without the enrichment. And then that goes straight to 10x genomics, or we run it on single cell plates with index sorting. And we collected from a range of around 4 post-conception weeks within the yolk sac up to 17 post-conception weeks with liver. And from this, we've got a cohort of around 214,000 cells across these various different tissues we looked at. Within the liver alone, we had around 145,000 cells, and these are the cell states that we found. So we've got a lot of progenitors, a lot of immune populations. The majority of it is erythroid, and then we're getting what you'd expect from endothelial cells, hepatocytes, fibroblasts, etc. One of the first things we do after we've clustered it to figure out what cells we've got, we base that a lot on the differentially expressed genes. So one of the things that we put together with this is just, these are some of the key genes that told us what are different. We look at a lot more than this. We generate lists of hundreds to go through. But this is a list of genes that really helped us tease it apart. And from this, we could also pull out some of the genes we know are expressed on the surface so that we can then do validation. So based on this, we put a panel together to sort out, almost 30 odd different types of cells for validation, so that we were able to look at things like the morphology. So these are just some of the cells that we have. So we were able to, based on the single cell transcriptome data, able to pull out some of the nice genes that will help us find these cells and see what they look like and what they might be able to do. We also wanted to look at the cells in space, so within the tissue, and this is looking at liver with dendritic cells. So you can see a bile duct in the middle, and this is using Hyperion data, so it's the mass cytometry, but in space. So it was really nice to be able to then, we use those markers to also look at what the arrangement of the cells might be as to how they could be interacting. One of the other things we really wanted to look at was the trajectory of these cells. So because we're going from hematopoietic stem cells through to some later stage cells, is what were we seeing in, what were the changes that happened, how did these cells progress, were they part of the same lineage and things like that. And we look at that with pseudotime. One of the methods is looking at an FDG. So this is all of the cells and how it splays out in time and how they're related to each other. It's a nice graphic, but the main thing is how it all ends together and how they're all linked and what that association means for development. And then when we zoom in on the hematopoietic cells, we see that we've got 3 main branches. So we've got the MEM branch, which is the megakaryocytes, mast cells, and erythroids. They form one main branch down the middle. We also have the lymphoid, and we've got the myeloid lineage that stem out from different points. Zooming in on the MEM branch, what we found, so stereotypically it's known that megakaryocytes and erythroids share a progenitor, but what we found is that the mast cells were also very closely related. So they're joined by this common progenitor in the middle, and I think it's something that has been noted in mouse but hadn't yet been shown in human, that there was this common progenitor for the 3 different cell types. When we looked at this within the non-lymphoid tissue, and also in the yolk sac, we actually saw quite a bit of that happening in the non-lymphoid, specifically in skin. So we looked into that a bit further, and what we found is that there are actually quite a few nucleated erythroid cells within skin. So we have that, part of that erythropoiesis is occurring outside of the liver. And we do know there are conditions where if this goes wrong, you get erythropoiesis continues to occur after birth. So there are conditions where it has gone wrong. So it is possible that this is a normal process that happens, and at some stage this stops and is then retained within the tissues that should be producing it. So we not only see these cells within vessels, but we're also finding these outside within the tissue. When we look at the lymphoid lineages, one thing that we found was really important, and if you look at the left, we've split the liver into 4 stages, is a link between the hematopoietic stem cells and the NK and ILC lineage we only see very early on in development. So I don't know if you can see it there. We've got an early lymphoid cell, and it just really, I guess, it highlights the importance of, if you don't split it up enough, if you look at it as a whole, you don't always see the connections that could be there. We really needed to look at the data by, like, within stages or subject to subject, but with the power of cells to be able to see some of the connections that might be there, that otherwise it appears like there's no link at all. And then we found a whole lot of these cells within the non-lymphoid tissues as well. We spent a bit more time looking at the B-cell differentiation because it was a cleaner system to look at. And what was nice is that we saw the progression from a hematopoietic stem cell through to pre-pro B cells, pro B cells, B cells, into final. And so we've got, like, we could see it within the morphology as well, and then you can see that there are changes occurring within the genes, and marked and bold are transcription factors. So we could see that there were these, like, key changes occurring as the development went on. Another thing we looked at, because we were interested in looking at liver compared to the non-lymphoid tissues, is when do you start getting a tissue-specific signature and where do you see it in the tissues? So in plenty of cells, the tissue, regardless of where it was the same cell. So mast cells, for example, look the same wherever they were. The biggest difference we see is really with the macrophages, not too surprisingly, where Kupffer cell in the liver, it's got VCAM, it's specialised for being where it is, whereas in the skin it's got factor 13A. So we do see differences. Inherently, it's a macrophage. It still has all of those same genes, but you know, it's got its own little flavours. And we also saw it a bit with the dendritic cells, where by the time it gets to the non-lymphoid tissue, so by the time it's reached the skin, it's a bit more mature. So you've got, I guess, a bit of an earlier state, and then it's a bit more activated by the time it gets out. And then, like, going back, I guess, to the origin, when we look at the HSCs and the multipotent progenitors, we found if we zoomed in onto the HSCs, we saw quite a bit of diversity within them. So this is just really zooming in on the main branches that come out, whether it goes to a B cell, a nute myeloid progenitor, which then stems to the rest of the myeloids, or to the MEMP. You can see that just within the HSC, when you really zoom in on it, it starts to get the flavour of the particular cells. So right back at the start, it's HSC through and through, and the rest of these HSCs as well, but you can see that they're starting to gain genes to become an MEMP, or starting to get them to become a pre-pro B cell or a new myeloid progenitor. So they're starting to get that commitment despite the fact that they're still a stem cell. And in wanting to look at their differentiation potential, we pulled them out. So we pulled out already committed, so not stem cells, and the 38 positive, and also pulled out HSCs, and single cell sorted these into plates, which were used for SMART-seq sequencing, and also for culture. So here we can see when we did the SMART-seq that it overlaid really nicely onto our 10x data. So we know that what we were pulling out into the plates that we also cultured were these stem cells. They were what we were expecting to find. And what we found when we did the differentiation cultures as to what they could become, so this is like colony forming assays, that we found that depending on the age of the stem cells or the sample that we got the stem cells from, they had different differentiation potential. So very young HSCs are less committed. They could become multiple different cells when you cultured them. So you'd find that they become myeloid and lymphoid, or granulocytes. But then as it moved on, there were fewer things that those HSCs would become. So they'd already gotten a switch, like somewhere during development, that they could only become one of a number of things. And you can see that as well, that just through time, you start getting less erythropoiesis from the hematopoietic stem cells. So it's not shown here, but we saw more myeloid cells coming up. So they're changing through time, depending on the cells that are needed during that stage of development, you get a change in what the HSC is actually able to do. And then you also get an increase in the B cells. So one thing in the lymphoid that I didn't mention is you don't get mature B cells until around 12 post-conception weeks, and that's actually reflected in whether the HSCs can make them at all. So what signals are occurring that mean this is still before the bone marrow is producing? So very early on, HSCs won't make a B cell, but then later on they can. So there are intrinsic factors within these HSCs that determine what they're able to become and how that fits with what's needed during development. So just to summarise, we've got an extensive resource of blood and immune development over gestation, both within the liver and then looking at across various other tissues. We've found a new, mast cell trajectory, and that comes from this common precursor within the erythroid and megakaryocyte lineage. We found that there's extrahepatic erythropoiesis, so it's not surprising to find it within the liver or within the spleen and things like that, but that actually we're getting some of that occurring in the skin. So how much more of these processes are occurring outside what's stereotypically known as a hematopoietic tissue, I guess. We nicely show the B cell development within the liver and how various different cells, specifically the myeloid, are adapted to the tissues that they're in. And then that, just as I lastly showed, the HSC multipotent progenitors have different potential over time, and that, this is an intrinsic change within them as to what they might be able to become. As I mentioned, this is all part of the Human Cell Atlas, specifically the Development Cell Atlas. So all of this is available online through their resource. We have all of it available in a portal, so if you went to the website, you'd be able to explore the data. This is the liver data. So you can go and you can look through it by stage, by cell type. It allows you to explore it by different genes that you might be interested. So the whole array of genes that we picked up by 10x are displayed on here, and you can see how that compares. So it allows people to do their own comparisons. There's only so much we can do ourselves, so anyone, depending on what their research, will have their own interests, and so there might be something that you've thought of that we weren't able to explore at this stage. We also have it in a more comparative format where you can look at multiple numbers of genes at the same time across whatever cells you want to look at. So this is a heat map in the form of spots, and we also have it in a more typical heat map. So this is just really nice to look at. One gene on its own does not tell you something, but you can look at a whole array, perhaps genes that are all associated with a pathway, and see how it might be changing through it. This was a big collaborative effort, so most of it was run within our lab, and we did it with HDBR, but Sanger helped a lot with some of the processing and things of the data, and we had quite a lot of funding and couldn't have done it without the core facilities at Newcastle. So thank you very much.